Hi everyone, my name is Katya and I'm going to, actually I will start with telling everyone that from today, VIPAT is no longer VIPAT. We are now called Synovis, officially rebranded to Synovis. I'm a team lead for the AML MRD section at Guy's Hospital. So I will tell you a bit about the real life in a diagnostic lab and how we do the monitoring for PML-RARA and NPM1 mutation. We'll stop a bit on the assays, internal quality assessment, and at the end we'll just do a few points of comparison between the CML and AML MRD. So acute promyelocytic leukemia is the most malignant form of AML, and if left untreated, patient dies within a month. Luckily, at the moment, there is 80 to 90% cure rate with the treatment of ATRA and ATU. The main genetic event in the APL is a fusion oncogene PML-RARA, which is formed after translocation of 15 between the long arms of chromosomes 15 and 17. Just very quickly, other than PML-RARA, in 1 to 2% of the cases with APL, there are all these different types of fusion transcripts. And currently in our lab, we also do monitoring for this one here, which is a translocation 1117. So currently, on the RARA I'll tell you a bit about the structure of the protein, of the gene, sorry, the fusion gene. So currently, the breakpoint on the RARA is somewhere in intron 2. And on the PML gene, there is a variable breakpoint. The most common one is it's in intron 6, which gives formation of a BCR1 subtypes, and this is seen in around 55% of the cases. In some cases, the breakpoint can be actually at the end of the exon 6, and this is forming a BCR2 subtype, and it's formed in around, and this is seen in around small proportion, 5% of the cases. And sometimes the breakpoint is in intron 3, which forms a BCR3, and that's seen in around 40% of the cases. There are few cases as well where the breakpoint can be in intron 4. And then the exon 4 is also part of the fusion protein. When a diagnostic sample comes to our lab, the first thing we do, we test it for PML-RARA 1, 2, and 3, and for the intron 4. And as well, we do test for the reciprocal RARA PML gene, which are seen in approximately 70% of the cases. They're expressed at a higher level, which afford greater sensitivity for the monitoring. Therefore, screening for both of them is actually recommended, for both the PML-RARA and the RARA PML is actually recommended. So we test, so as I said, first we do the testing for the diagnostic sample and we assess which is the correct breakpoint, and further patient is monitored by only the correct assay. So just a bit for the samples that we require a sample, a bone marrow sample. After we test, actually, we require peripheral blood and a bone marrow sample, but we test only the bone marrow sample after each course of chemo, and then for every 3 months for 2 to 3 years. So peripheral bloods, we also always kind of request, but we don't test them unless the bone marrow fails. Then we have a transplant. If the patient, however, goes to transplant, we require samples at day 0, 30, 60, and 100. And here the crucial samples are obviously the diagnostic one, and then the end of treatment, where for optimal result and the patient have a low risk of relapse, they should be in undetectable levels of PML-RARA. Moving on to the NPM1. So the wild type NPM1 protein has all this function, and that's a gene, and the gene for the NPM1 is very often mutated, and it's mutated in around 20 to 30% of all AML cases. Usually it's associated with a good prognosis, but it depends on co-occurring of other mutations, obviously. The mutations are usually are always an insertions of 4 nucleotides at the C terminal, which lead to a frame shift, which result in changing of the last 7 amino acids of the protein with 11 different residues. As a result, there is a functionally stronger nuclear export than import signal, which is contrary to the wild type NPM1, and consequently, there is a cytoplasmic location of the protein. In 70% of the cases, the insertion of the 4 nucleotides is at this position here, and they always DCTG, and this is known as a mutation A, and in another 15%, there is a mutation B or D, which have these 4 nucleotides inserted at the same position. As with the APLs, similar to the APLs, we ask for samples after each course of chemotherapy, and we do test the bone marrows. Again, and most crucial is the peripheral blood at post-course 2, which is proven that if negative, if the NPM1 variant at this point is undetectable, then the patient has a very low risk of relapse further. Again, if patient goes to transplant, we want samples at day 0, 30, 60, 100, and then is proceed with the same monitoring. We always need a diagnostic sample for these patients, and we need to do sequencing, Sanger or NGS, to assess for the correct mutation type. So in a lot of the cases, we do have, well, as I told you already about the A, B, and D mutation, but there is, at the moment, I think there are around over 100 NPM1 variants described in the literature. And in our lab only, we have over 55 and counting. That's just a sample of all the different NPM1 rare type of mutations we have in the lab. As you can see, there can be different nucleotides inserted in the same place, or they can be connected with deletion or insertion, but they all lead to the same frame shift. So when a new NPM1 mutation that we do not have an assay currently in the lab comes to the lab, we usually do design a new assay. We use the same forward and NPM1 forward primer and an NPM1 probe for all our rare variants. And we do design only the specific NPM1 reverse primer, which covers the NPM1 variant. When designing the primers, we do look at all these. Length of the primer need to be around 20 base pairs, do not have strong self-dimers or heterodimers, melting temperature around 60, and GC content about 40 and 60%. We also design G blocks, which are small double-stranded synthetical DNAs, and we dilute them and use them as a standard curve. After this, we have, when the primers arrive, we test the 3 sort of separate assays that we've done, and using the G block as a standard curve. And then we choose the best one based on Y-intercept, which should be between 38.5 and 40 R-square, efficiency of the reaction, which need to be between 90 and 110, and there should be no amplification of the normal blood control. Then we do acceptance into use and create a monitoring form. Talking about the monitoring form and the internal quality that we've designed for each assay, I'll show you a bit about the quality assessments that we have in place currently in the lab. So this is a picture which shows all the kind of main steps where which each sample goes through, and we did put a control at each step, and each control is tested. So here we have the RLT solution and the normal blood control, which is tested throughout, kind of go through the whole process, and is tested at the qPCR. Each plate do have an MBC on it to assess kind of the to assess for any contamination. So far, we found that this here is probably the most sensitive part, and probably because it's still manual and we are currently trying to automate it. And then I put here high and low level controls because these are something that we are thinking to implement in the near future. For each run, a front sheet is filled where everything is recorded, from date and run number to all the checks done, who's done the QC checking of the plate, and who's reported it. The person who's doing the QC is doing a assessment. So first of all, they record all this data on a separate IQC sheet, and then they they look at, especially, so we monitor the intercept standard 2 and standard 4 a bit more closely, and we create the Levy-Jennings graphs for them, and then based on the result from here, following the Westcott rules, the QCer is actually accepting the plate for passing the QC, and it's fine to be reported further. Recording all the batch numbers, the machine that the qPCR was done is good because this we are able as well to monitor variations between different batches of reagents and standards used. During the QC of the plate, the person who is performing the QC is also assessing the sample quality. So they look at all the samples, and based on the ABL CT values or copy number, depending, they can decide if the sample is optimal and is good to go for reporting, or sample has suboptimal quality, and if there is no obvious reason why, they do activate it for repeat extraction, so that we kind of reduce the time of reporting and don't delay the sample unnecessarily. A sample is accepted to be failed when the able copy and the able CT is above 30, or the copy number is less than 999, and repeat extraction is always requested along with the extraction of the PB, so the PB can be also tested. Another type of samples that need re-extraction are the unexpected positive samples. If there is a sample which shows positive result with the previous undetectable trend, first we ensure that the sample is real positive by re-extracting it, going back to the RLT and going through the whole process again. And then we follow this algorithm here. And I'm not going to go in detail in the reporting further because I'm sure that Dr. Dillon will go through a bit later on. So further, we also look at the same intercept standard 2 and standard 4 for kind of long-term trend by creating this monthly monitoring report, which looks at the average value of the CT for the 6 months running. We also record on a monthly basis the failure runs, the failed controls, make a comments about any kappas, and also record total number of assays. And as well, at the end, we accept the kind of the assay for the month if it had a satisfactory or unsatisfactory performance. So our assays, we would like to think they're really perfect, but there is always something to change and improve. There is always room for improvement. And for this, for any small change we need to do, we also fill a change control form, which focus on first asking the question, what is the change? Do we need the change? And this is also true for, do we need to introduce a new test or not? And it goes through a validation where we test a minimum of 100 samples with known results. We assess reproducibility, repeatability, limit of detection. And then we look at the IQC and the controls. Are the controls that, if it's a change in the process, are the controls still acceptable? If it's no change, but it's a new process, what controls should be put in place for the internal to kind of perform an internal quality control? We look also at the EQAs. Is there anything available for NECOS? If there isn't, we look at laboratory exchange of samples and kind of ensure this comparability. Also, obviously, the SOP needs to be updated and training needs to be performed with everyone who need it. So in the last couple of minutes before I give the before we go to Nikki, I'll just do quick comparison on few points between the current state of the MRD monitoring between the CML and the AML. So to start with a few of the similarities, both are done on RT-qPCR. Both are using ABO1 gene as a control gene in most of the cases. However, GASB is also used. And then from here, there are, I think, more differences than similarities. So to start with the standards available, currently in the AML, we use for the main kind of well-known mutations, like a mutation, the type A or the BCR1 and 2. We do use the company standards, but not which are not internationally used, and they, depending on the company, they might have a different performance, which is in comparison with the CML, where there is a really nice calibrated World Health Organization reference panel, which we've heard a lot in the morning. Another thing is the use of the MRD, so it's slightly different between the CML and the AML. I've already mentioned about the AML, and in the CML as well, what we look at is at 3 months, patient needs to, for optimal performance, patient needs to have less than 10%, at 6 months less than 1%, and at 12 months should be 12 months and further, patients should have 0.01%. Also in AML, as I've already mentioned, we do stop sort of monitoring if patient is negative for 2 to 3 years after the treatment. As in CML, this might be going for, in most of the cases, it does go for life. The samples that are recommended for AML are to use for testing is the cDNA-B bone marrow, and for the CML, it's the peripheral blood. It's going to be okay. And then the way the results are presented is slightly different. So in the CML, there is this international scale ratio, which allows for uniform testing and result comparison between different labs. As with the AML, we do present the result as a fusion or a mutated gene copies in 100,000 able copies. And I'm really sorry about, I don't know what happened. I've just looked at this presentation, was looking much better 10 minutes ago. And with this, I want to thank you for your attention. And I want to thank to the Synovis Cancer Genetics team, especially to Kathy, Mary, Shadia, and the whole of the admin team who are at the core of the AML MRD. And Nicola and Richard, who are our leads, and as well the trial lab. And with this, I'll give the word to Nikki. Thank you.
So thank you to the organisers for the invitation to talk to you today. So I'm following on from Katya, and I'm actually going to be talking about CBF leukemias, so core binding factor leukemias, so RUNX1, RUNX1 T1 fusions, CBFB and MYH11, and then the rare fusions. So the presentation focuses on real-time qPCR, and I've made the assumption that many of you will have knowledge and understanding of the markers and the relevance to AML. Richard is also going to touch on that later. So I wanted to break this down further into the frequency of the trackable mutations in age-specific cohorts, as you can see here. So PML-RARA and NPM1, just described by Katya, represented by blue and orange segments. So the orange being and NPM1 being more frequent in the adult populations. And then the CBFs, so 821 in red and inversion 16 in green, being more common in the children. You can also see the grey portion of these pie charts, which indicates the percentage of patients without trackable markers by real-time qPCR. And this is where we focus a lot of our time and efforts in Richard Dillon's trials lab. So this is a combination of needing to identify trackable markers and design assays for monitoring, and patients not having trackable markers, but the vast majority of these we think we can find markers for. I've moved my slides around. There we are. So let's start by discussing briefly the assays for core binding factor leukemias. So these were described in 2003 as part of the European Against Cancer programme and form the basis for our ELN current guidelines. So the 821 fusion, which you can see at the top here, is conserved in all patients. So we only have one assay, forward probe and reverse shown here on the right. But for the inversion 16s, there is a common fusion occurring in 88% of patients that we refer to as type A, between exon 5 and exon 12. And then we have a fusion that we refer to as D, and then another in E, occurring in 5% of patients that have different fusion breakpoints and therefore require different assays, as shown here. These are in the current ELN guidelines, but it's important to recognize that there are actually an additional 7 breakpoints in this gene, very rare, described in 1999. And we have actually validated assays and have been monitoring various patients with these rare fusions in AML 17 and AML 19, so the National AML Trials. So Katya has touched on what's required for the real-time qPCR assay tests and shown you how they should be carried out in the lab. So I'm not going to touch on that, but the assays in the ELN guidelines, so 821 and version 16 A, D, and E do have commercially available standards, which we use from Ipsogen. But the other assays that we have for the rare fusions, we do not have standards for those. And that's something we're working on in the lab. Suffice to say, all criteria that surrounds a test need to be met for the result to be valid. So again, Katya's described how often samples should be collected. The baseline sample, preferably a bone marrow, is crucial for inversion 16 so that we can identify the breakpoint and attribute the correct assay to the patient. As there is only one assay for the 821 fusion, there are exceptions where we have monitored without a baseline sample, but these are very rare and at Richard's discretion. How often should the samples be collected? Well, again, Katya described that as she had done before. Apologies. So what are the additional reporting considerations that we need to look at when we're talking about core binding factor leukemias? Well, there's a significant proportion of patients that do not achieve PCR negativity, and they demonstrate low-level transcript expression at the end of treatment, but it's not predictive of relapse. And so we follow in our lab, and this is just transferred into now it's in the lab downstairs, the use of what we refer to as yin thresholds that were published back in 2012. And this is a threshold that is indicative of relapse. So we're looking for disease level to exceed the threshold in the peripheral blood or bone marrow before it's indicative of relapse, and clinical action needs to be taken. So for the inversion 16, we're looking at greater than 10 copies in the peripheral blood, 50 copies in the bone marrow. For 821, 100 copies in the peripheral blood and 500 copies in the bone marrow. At the end of treatment, if a patient does test positive, but below the threshold, this should be described as molecular persistence at low copy number. If this is the case, then the recommendation is to test every 2 months to ensure that the expression level is stable and below the threshold. So I wanted to quickly go through some real life data with you to get a feel for what we're looking at with regard to the CBF patients. This, for those of you who are unfamiliar, or I doubt there are many of you, is an MRD profile. Bone marrow results represented in red, peripheral blood you can see here in blue, open shapes or white shapes are negative results, and the colored shapes are positive results. So this is a relapse in a version 16A patient that was confirmed by a rapid repeat. Patient did reach complete remission, and so the relapse was clear. We also see in these patients what we refer to as a rapid relapse. So although the patient is following recommended sampling every 3 months, even within that window, we can receive a sample and test, and the disease level will be comparable to diagnosis. So there is some discussion around whether or not some patients should be monitored every 2 months to try and capture these rapid relapses before they're at the level of diagnosis in the patient. This was a frank relapse, so no rapid repeat sample was necessary. Patient responded and eventually returned to complete remission. So this is one of the cases where we see continuous low-level positivity in a patient, and we call them grumbling MRD profiles. I've indicated the threshold, the yin threshold here for you, and you can see that this patient was monitored over several years and did not relapse, so standard of care monitoring. This is a more complicated profile to interpret. It's still a grumbling profile, but you can see that every so often one of these regular monitoring samples is perhaps around the level of the threshold. A rapid repeat sample and the disease level returns below the threshold. So difficult to interpret, and monitoring in the bone marrow frequently is absolutely essential. In this patient, continuous bone marrows were not satisfactory for the patient, I think, over time, and so more rapid MRD monitoring within the peripheral blood was carried out. So I'd like to return to the patients with some of the rarer markers and with no marker. And we've been monitoring the rare translocations in our lab, usually detected by FISH or more recently targeted panels. So in this case, we're talking about NUP98 NSD1 fusions or 511 translocations, DECCAN on 6/9 with 6/9 translocations, and then the more complicated and very diverse group of MLL fusions, so KMT2A. There's no commercially available standards for these, except in some of the more common MLL cases, but they don't seem to perform in our hands as they do as our other standards, so for 821 NPM. So we actually don't use these ones, and it's something that we're looking at in the lab. I wanted to sort of backtrack a little bit and go over why we do monitor some of these rarer translocations, because we were able to collect data in collaboration with a variety of different clinicians. This is unpublished by Adam Ivy, to allow us to monitor the patients serially and predict outcome where the rarer leukemic fusions were present. So you can see the fusions here with the profiles represented by various different colors. In some cases, we were able to monitor patients that remained in complete remission. And this was obviously communicated with the clinicians who saw the same thing. We were also able to identify relapses. In this particular MRD profile set, you can see patients did reach complete remission, but we found molecular relapse, shared that with the clinician, who then didn't intervene with any treatments until frank relapse was confirmed rather. And you can see that in all of these cases, disease levels did return to that found at diagnosis. In some cases, we were able to detect ongoing low-level disease, not identified by more traditional methods, which again ultimately returned to levels comparable to that at diagnosis.
2 minutes over
So
now, Nicola, that's all right.
Sorry?
2 minutes over time now, so if you could bring it together soon, sorry.
Okay, In which case I will skip a couple of slides. One of the other things that we do in our lab is to identify the rarer fusions by true site RNA fusion panels. This allows us to pick up really rare translocations and design assays. One of the cases that we've been able to detect was a SMACA2 ETV6 fusion confirmed by the reciprocal. Pop an assay in there. We have a lot of criteria that we use when we're looking at these rarer fusions, because we don't have the luxury of any sort of commercially available standards. So we have to go back to basics with monitoring and checking that the assays are performing correctly. So no positivity in normal samples. One of the crucial things that we do like to see is that the marker CT is comparable to the ABL control gene at diagnosis. And that gives us an indication that we're looking at clonal mutations, which is, I think, up for maybe discussion a little bit later. This was the profile that we monitored in the patient. So patient initially responding to treatment, went into remission. Relapse was very clear, but not acted upon until it was frank. A patient has then responded to treatment and is responding still. So just very quickly, we've adopted this approach for the MyChild trial to try and identify rare markers that we could monitor. We've completed this in 184 patients, and in a third of patients, we were able to monitor rare fusions. In a third of patients, there were no fusions, but in the outstanding third, the RNA-seq approach allowed us to modify and improve the assays that we already had. So thank you to the clinicians and patients involved. And this is the team with Richard at Guy's. Thank you.